In a museum drawer in Paris, sealed inside a block of amber the color of dark honey, there is a scorpion. It has been there for 100 million years. Its claws are still raised. Its tail still curves over its back in the same fret posture a scorpion takes today when something comes too close. It looks, with unsettling precision, like the scorpion you might find under a rock in your backyard tonight. That sameness is the first clue that something extraordinary happened here. Something that most of the science built around this animal had quietly gotten wrong. For most of the 20th century, the story of the scorpion felt settled. Ancient, yes. Dangerous, certainly, but understood. The textbooks placed their origin in the sea, a straightforward march from ocean to land, a single lineage that crawled out of the shallow Silurian waters around 430 million years ago and never looked back. One origin, one transition, one clean line from water to desert sand. We were wrong about almost all of it. Not about the age. The scorpion is genuinely one of the oldest animal body plans on Earth. Older than the dinosaurs by hundreds of millions of years. Older than trees. Older than the wings of insects. But the story of how it got from that Silurian seafloor to the inside of your shoe is stranger, more tangled, and more violent than anyone imagined. And most of that story was invisible until scientists learned to read it in the DNA of living scorpions. Not just the big ones you picture when you hear the word. All of them. The 2,500 known species spread across every continent except Antarctica. The ones that glow blue-green under ultraviolet light for reasons nobody fully understands. The ones that can slow their metabolism to almost nothing and survive a nuclear test site. The ones whose venom has been sitting in a molecular arms race with prey animals for so long that it has independently evolved the same chemical solutions on opposite sides of the planet. This is what the DNA actually shows. Before we get into it, hit that subscribe button. Every video here digs up the real origin of something you thought you knew. Let's begin. The old story had a satisfying shape to it. Scorpions, the textbook said, evolved in the sea during the Silurian period, roughly 430 million years ago. These early marine scorpions, relatives of the fearsome sea scorpions called eurypterids, gradually adapted to life in shallow coastal waters and tidal zones. Then, at some point, they made the crossing onto land, and from that single terrestrial ancestor, all modern scorpions descended. The evidence seemed solid enough. There were fossils. There were the eurypterids themselves, giant water-going arthropods with the same basic body plan, the same segmented tail, the same claw-bearing arms, that confirmed scorpions had deep marine roots. And there was the morphology of early scorpion fossils, animals with gill-like structures that suggested a body still designed for water, still carrying the memory of the sea in its anatomy. The logic was clean. The sea gave birth to the scorpion, the land received it, and from that single landing, 2,500 species radiated outward across 430 million years. But the fossils had a problem, and the problem was the gaps. The scorpion fossil record is rich at its oldest end and rich in the modern era, but the middle, the critical window when the animal was supposedly making its transition from coastal wader to fully terrestrial predator, is almost empty. The rocks simply did not preserve what happened, and for decades, researchers filled that gap with assumptions. Reasonable ones, carefully argued, but assumptions nonetheless. Then the genomes of living scorpions became readable. And what they revealed did not just fill the gap. It blew the gap open into something far more interesting. In 2022, a landmark study led by Prashant Sharma at the University of Wisconsin-Madison, working with a team of collaborators across multiple continents, published the most comprehensive genomic analysis of scorpion evolution ever attempted. The team sequenced and compared the genomes of scorpion species drawn from every major family across the globe, anchoring their genetic trees against the fossil record using a technique called molecular clock analysis, which measures how fast DNA mutates to estimate when lineages split apart. The scale of the data was staggering. Hundreds of genes across dozens of species, mapped against 450 million years of evolutionary time. And what the data showed cracked the single origin story open at its foundation. The first revelation was the timing. The molecular clock pushed the origin of scorpions deeper than anyone expected. not 430 million years ago, but closer to 450 million, placing the earliest scorpion ancestors in the late Ordovician, before the great Silurian radiation of sea life that the fossils had pointed to. This mattered enormously because the Ordovician ended in one of the 5 great mass extinctions in Earth's history. The scorpion lineage, the data suggested, had already existed before that catastrophe and survived it. The animal that would one day live under rocks in the Sonoran Desert had already been alive when most complex life on Earth was being erased. The second revelation was stranger. The transition from sea to land. The single crossing that the textbook story depended on may not have been a single crossing at all. The Sharma team's genetic tree showed deep, ancient divisions between scorpion lineages that could not be explained by a single terrestrial ancestor radiating outward. The splits between the major scorpion families were too old, too deep, and in some cases too geographically isolated to fit the model of one ancestral population coming ashore and then spreading. The data pointed instead toward multiple independent colonizations of land. Separate lineages making the crossing from aquatic or semi-aquatic existence to fully terrestrial life at different times and in different places. Not one scorpion dragging itself onto the beach and founding a dynasty, but several lineages across a span of millions of years, each finding its own path from water to stone. This is the same pattern DNA has revealed again and again in the deep history of life. The clean single origin, the one bottleneck moment when a thing became itself, is almost never as clean as it looks in the fossil record. Life is messier than that. It finds the same solution multiple times, from multiple directions, because the same pressures produce the same answers when the raw material is similar enough. The scorpion, it turned out, was a master of that redundancy. It had solved the problem of living on land not once, but repeatedly, across geological time, in different parts of the world. But the third revelation is the one that carries the deepest weight, and it concerns the scorpion's most famous feature. The venom. Scorpion venom is not one thing. This is the part that stunned the researchers most completely. When the team mapped the genetic origins of the toxin-producing genes across their tree of life, they found something that should have been impossible under the single origin model. The same classes of venom compounds, the same molecular weapons, had evolved independently in scorpion lineages that had been separated from each other for hundreds of millions of years. The potassium channel toxins that make the venom of the fat-tailed scorpion of North Africa so dangerous to the human nervous system are structurally similar to the toxins in the venom of the bark scorpion of Arizona. But those two animals last shared a common ancestor before the Atlantic Ocean existed, before the continents had pulled apart, before most of the geological features of the modern world had taken shape. They did not inherit their weapons from a shared ancestor. They built the same weapons independently, from the same raw genetic material, or the same evolutionary pressure, on opposite sides of a world that was once joined, and is now an ocean apart. This is what biologists call convergent evolution, and in scorpions, it runs so deep that it rewrites what we thought we knew about the venom itself. For decades, researchers studied scorpion venom as though it were a single system, a coherent pharmacological toolkit passed down from one ancestral species. The DNA revealed it as something far more unnerving, a set of solutions that life keeps arriving at on its own, because those solutions work, because the prey animals they target have nervous systems built on the same ancient biochemical architecture, and the fastest way to stop a nervous system has not changed in 400 million years. The scorpion, in other words, did not just survive the eons unchanged. It kept reinventing its most dangerous feature from scratch, over and over, because evolution kept finding the same answer. Now comes the part of the story nobody talks about, the part hidden not in what scorpions became, but in what scorpions lost. The fossil record of early scorpions shows something remarkable. The first scorpions were enormous. Some of the Silurian and Devonian species reached lengths of nearly a meter. They were apex predators of their shallow coastal environments, built on a scale that modern scorpions cannot approach. The largest living scorpion today, the giant forest scorpion of South Asia, reaches about 23 centimeters. The emperor scorpion of West Africa, famous as a pet trade staple, barely exceeds 20 centimeters. The giants are gone, and the DNA reveals why, in a pattern that mirrors every other story of domestication and narrowing and loss on this channel. The ancient marine scorpions were genetically diverse, varied across their range, experimenting with size and form in the way that only ancient, abundant, ecologically dominant animals can. The transition to land, whether it happened once or multiple times, imposed a bottleneck. Moving onto land meant abandoning the buoyancy of water, the thermal buffering of the sea, the abundance of marine prey. Only certain body plans survived the crossing. Only certain sizes worked in the new environment. The diversity of form that the ancient sea-going scorpions represented was largely erased. What we see today in those 2,500 species is not the full range of what scorpions once were. It is the narrow surviving slice of a much larger, much stranger experiment. The glowing, the venom dripping, the heat-tolerating animals that crawl through the world's deserts and jungles and steppes are the remnant of something that once dominated coastal seas and forms we can barely reconstruct from fragmentary fossils and the ghost signals left in living genomes. There is one more layer to this story, and it is the one that makes the amber scorpion in that Paris drawer so unsettling to look at, because the scorpion body plan is not just old. It is, by the standards of evolution, almost frozen. When researchers compared the genomes of modern scorpions to the inferred genomics of their ancient ancestors, they found something that researchers had glimpsed in the morphology, but had never been able to confirm at the molecular level. The rate of evolutionary change in the scorpion body plan is extraordinarily slow. Not because scorpions stopped changing their DNA. They didn't. Their venom genes churn with variation, locked in an arms race with prey so relentless that new toxin variants appear across geological blinks of an eye. But the underlying body, the 8 legs, the 2 claws, the segmented tail ending in the sting, the book lungs that allow them to breathe, the sensory hairs that detect vibration through solid ground with a sensitivity that can feel a beetle walking 3 feet away in the dark. That architecture has remained essentially stable for 430 million years. The scorpion found a body plan so effective that evolution almost never improved on it. Not because nothing tried to compete. The history of life is full of animals that must have preyed on scorpions, competed with them, tried to displace them, and yet here they are, in the Sahara and the Atacama and the Himalayas and the rainforests of Borneo and under the rocks of suburban Arizona, everywhere. Still, the stability itself is the message. What does this mean for the scorpion under the rock in your garden, or the one in the museum, amber, or the one in the ultraviolet light, glowing with colors no eye was ever evolved to see, for reasons the DNA has not yet told us? It means you are looking at one of the oldest continuous experiments in animal life on this planet. An experiment that began before fish had jaws, before plants had conquered the land, before the oxygen levels in the atmosphere had stabilized enough to reliably support large animal bodies. The scorpion did not survive and changed because it was lucky. It survived because it built itself around solutions so deep and so robust that the eons have not found a reason to discard them. Multiple invasions of the land. Venom rebuilt independently from scratch on opposite sides of the world. A body plan so successful it calcified into permanence, while everything around it changed beyond recognition. And beneath all of that stability, a history of losses we are only now beginning to read. The giant forms gone, the marine diversity erased, the strange transitional animals of the Devonian and Carboniferous, preserved only as fragments, shadows, genome ghosts in the living species that outlasted them 430 million years ago. Something that was not quite yet a scorpion pulled itself through shallow coastal water, felt the hardness of stone beneath its legs, and did not go back. We do not know exactly when. We do not know how many times that crossing happened, in how many places, in how many generations of creatures trying and failing before one lineage held. We only know that it worked, that the experiment ran, that the body plan locked in and the venom kept reinventing itself, and the glowing began for reasons the DNA is still keeping to itself, and that 430 million years later, the scorpion is still here, still raising its claws, still curving its tail, still waiting in the dark for something to come close enough. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.